Okay, hopefully you all managed to get this plot for monocle using the Gaussian model. And you see that it performs not nearly as well as it did using the negative binomial model. So if you have access to counts, it's often better to use counts for differential expression methods that are based on models, at least. The last one that I'm going to have run is MAST, which is based on a zero-inflated negative binomial model, as opposed to monocle and edge R, which are just plain negative binomial models. And it uses something called a hurdle model to combine tests of discrete, so dropout versus not dropout, and continuous, so all the non-zero values, aspects of gene expression to detect changes in means. And again, uses a linear modeling framework like monocle and edge R, so you can incorporate batch information or other confounding factors that you want to control for if you want to. But MAST does not normalize their data, so you need to normalize it in log. No, you don't need to normalize it. You need to log transform it. MAST assumes log transform data. So here we've log transformed the counts because that's what this method specifically specifies. So again, you need to be careful when you run each method that you check what type of data it expects as the input. And be sure you're providing that. So yeah, we just read in the data, similar to the other ones, but again, slightly different object. And this method recommends when you use the model to add in as a confounding factor the total number of detected genes per cell, which is what that factor is, which we're defining here. And then here we do a log ratio test of the different GLM models. And then this gives it some very complicated output, but if you select the stuff called H, that's the hurdle model test. And we'll get you the p-values. For the differential expression. So here's the ROC curve. And you can see it performs very similarly to edge R and MAST and monocle in terms of AUC. So more complicated, but not necessarily getting you a better result. Then there are slow methods that, for time and convenience, we haven't run, but we provide you the code so that you can run them at home if you're interested. They take a long time to run and may or may not provide better results. So BPSC is based on the Poisson beta model. Again, uses linear modeling like MAST and monocle and edge R. So it can do all those things, but instead of using negative binomial models or zero-inflated negative binomial models, it uses a Poisson beta model. Yeah, so, and it, instead of doing group 1 versus group 2, it will do multiple groups versus a control group. So here you specify the control group, and then do your model design, and then do the GLM test. And you can see that this has the option to running it in parallel because it's very slow. So if you have a nice big computer core, you can run this in parallel and get results in a reasonable amount of time. The other method is, yep.
When you say may or may not be better, is that it actually depends on the data set, or is it just?
So we've only compared all these methods on one data set, And on that data set, this was not particularly better than a jar or the Wilcox test. Can't say that's going to be the case for other data sets. But in our experience, it's not better than the alternatives. But it is a lot slower. In contrast, there's SCDE, which in our experience is better than a jar and the alternatives, but not a lot better, but is definitely better, but is also extremely slow because it's based on Bayesian statistics to fit a zero-inflated negative binomial model, and it accounts for the relationship between the dropout rate and the mean expression and the variance in the mean expression in its model. So it's a very complete model, but it takes a long time to fit it and run the test. But it does, in our experience, at least for full transcript data, this does give slightly better results. Yes, it can be parallelized. So this is all code for running this. So there's lots of parameters here that will affect how accurate it is versus how long it takes to run. So you, again, look through the instructions for how to run this, and you can sometimes speed it up a bit. And this can be ran. So here, there's N cores, which will allow you to run it in parallel if you have the capability, the computing infrastructure to do that. So are there any questions on differential expression? Yep. For which method? So EdgeR uses a model and will do 2 groups, but you can specify which 2 groups, and doing multiple pairwise comparisons is significantly faster. MAST is 2 groups. All of these are specifically 2 groups, but except for BPSC, which will do multiple groups. But again, it's still doing pairwise comparisons. It's just sort of set up that it will do multiple groups against one control group more efficiently. Mm-hmm. In that case, you'd want to select those cells in each from your data set, and then create a new data set with just those cells, and then use these methods. So if there's no questions on that, we'll just summarize everything we've covered these 2 days as our ideal single-cell RNA-seq pipeline, at least in our opinion, as of this date. Of course, this is a developing field. We've already changed this from the last time we gave the course. It will probably be different the next time we give the course. But it will always be available on the web. So if you want to know our most recent suggestions, you can always go check. So experimental design is extremely important. If you can have input at that stage, try and make your voice heard. It's not always possible. But it's important to try and avoid confounding biological and batch effects. Many of the early single-cell RNA-seq data sets, they did one day, one condition, and another day, another condition. And it's just really impossible to get rid of the batch effects. So nobody knows whether the differences seen in those data sets are real biological effects or some technical artifact. But more recent experiments are getting much better now that this has come to light. So it's going to be much trickier to get your study published if you've got completely confounded batch effects now. Their unique molecular identifiers are really good at reducing the noise in your data, but they come at a cost. you completely lose any splicing information. It may reduce gene detection rates, although it's somewhat unclear because the methods, the protocols that use UMIs are different from the protocols that do not. So it might be a protocol difference rather than an effect of the UMI itself. It's better to use longer UMIs and then correct for sequencing errors than to use short UMIs and then have problems of collisions because those are harder to correct. And we recommend using UMI tools to correct for sequencing errors when you're counting UMIs. If you can't use UMIs, Spikeins may be helpful. They're definitely useful for quality control. And if you're interested in different cell sizes in your particular experiment, Spikeins are very useful for estimating the cell size. It's not always relevant for the experiment. Most of the data sets we've been talking about in the course, it's not relevant for, so it wouldn't have been very beneficial. Spikeins often exhibit higher noise than endogenous genes. This might be something to do with the Spikeins themselves, or it might be pipetting errors or degradation or some other problem along the way. Certainly, pipetting errors are known to be one contributing factor to the higher noise of Spikeins. So it's sort of, if you have someone who's a very good experimentalist, it might be worth doing Spikeins. If you're not a very skilled experimentalist, it might not be worth doing Spikeins. It also requires more sequencing depth. So if your budget for sequencing is very tight, it's probably not worth your time to put in Spikeins. Whereas if you have, if you're, say, at the Sanger and have access to tons of sequencing machines, and you can get a discount on your sequencing, it might be worth your time to do Spikeins. Cell number versus read depth is very, very dependent on what question you're trying to ask. So gene detection plateaus at about 1 million reads per cell. But in practice, because of the variance of the number of reads you get per cell in an experiment, that means to get 1 million reads per cell for most of your cells, you actually need an average of 3 to 5 million reads per cell. And then it starts plateauing. But if you're not interested in transcription factors or very lowly detected things and you just want to find cell populations, you don't need to detect every gene. You can. So you can get away with sort of hundreds of thousands of reads per cell, if that's all you're interested in. We're not specifically talking about UMIs. It's
less reads, if you're not interested, if you've only got a small amount of cDNA there to sequence in the first place. The longer the length, the more reads you need to cover it.
Yes. Yeah, if you want splicing, we're not talking about splicing information. If you want splicing information, then you want as many reads as you can. If you're just wanting to detect, get reasonable quantification of sort of transcription factors, you need about 1 million reads per cell. If you want splicing information, you want more.
Because transcription factors are low expression.
Yeah. So if you want to do regulatory networks, you need quite high-depth sequencing. If you want to do splicing, you need extremely high-depth sequencing. If you just want, if splicing is possible at all. Some people will argue splicing is not possible from single-cell RNA-seq data. So, but I've seen one paper that claims to have done it. So I'm reserving judgment, but you definitely need a lot of reads per cell if you want any hope of doing splicing. But if all you're interested in is detecting cell populations, you don't need that much. Yeah, so, and this is how to run an unconfounded experiment. So if you can do all your different conditions on one plate, then in theory you have no batch effects. If you do each of your conditions separately and you only do one replicate of each one, it's completely confounded, and your statistics are hopeless. There's nothing that can be done for that. If you do your conditions on the same plate, but across multiple different plates, you have batch effects, but you can correct for those statistically. And if you have replicates, you can correct for some batch effects and not others, so it's generally better to use sort of mixture models and models that can account for batch information as well for looking at those data sets. So technical replicates, I mean multiple biological replicates here. So you're doing multiple plates of the same cell population rather than trying to do a technical replicate from RNA from the same single cell. It is possible to do that. So you can lyse a cell, take the RNA, and then split it into two groups and do it in parallel. But then you're cutting your starting material in half. So you're losing more information than you're gaining in most cases. So I've only seen those types of experiments done for validating new sequencing methods and new protocol designs. Yeah. Usually do the whole cell and then do biological replicates. So you can think of each cell as a biological replicate, or you can think of each plate. So there's often plate effects. So the particular concentrations of the reagents you put in a particular plate or a particular experiment will vary slightly, and that can be detected. So you can get batch effects from just doing the same experiment at different times of the day or using different sets of the reagents, which are often referred to as replicates. So if you're doing the same biological condition on a day, that would be a replicate.
This is all geared to what you're talking about. This is in terms of using things like fluidine,
Yeah.
6-well plates, etc.
Yeah.
So if most people, well, a lot of people are getting towards doing the drop seq type.
It applies.
You can
then
avoid
it's
batches altogether, can't you? Maybe.
If you do it all in one run.
Yeah.
But if you're doing different runs on different days, you still have batch effects. So it could be more viewed as what day you're doing it on, as opposed to what particular thing. Plate.
If you're trying to achieve a certain, say, 8,000 cells or something is enough for your entire.
Yeah. If you can do it in one run on a 10x, that would count as that. Yeah. But depending on what you're looking at, it may or may not be possible to do that. So for processing reads, we recommend FastQC or FastQC for ReadQC and CutAdapt for trimming. For mapping, we recommend for small data sets or UMI data sets aligning to the genome or transcriptome using STAR. If you have a large data set without UMI tags, you can use Salmon, and it'll be much faster. For quantification, small data sets with no UMIs, we recommend feature counts, although HT-seq is essentially the same. There's not really a difference between them. For large data sets without UMIs, Salmon is a good choice. Again, just because of the speed, and for the benefit of the speed outweighs the loss of accuracy in most cases. If you have UMIs, we recommend UMI tools and then using feature counts to get the counts from that. For the expression matrix, we've been using Skater, so we obviously recommend that for QC. Currently, we recommend Scran for library size normalization, but there's been 2 or 3 new normalizations published recently, so we may be changing that soon. For batch correction, again, RUVS seems to work quite well, although that may also change as there's a review of batch corrections that we know people are working on at the moment. So we're going to bug them in a few months to see if they've found one that's better. For biological interpretation, feature selection, we recommend M3 drop. Clustering, SC3, pseudotime. If you have very distinct time points, so your cells cluster mostly, rather than being a smooth continuum, T-scan works well. If you don't know the number of branches, the only method that will try and figure out the number of branches itself is monocle version 2. But if you have a large continuous data set, Destiny seems to perform well, and you know the number of branches.
Is it possible to extract the pseudotime from just using the Destiny package?
Yes. Yeah. And then for differential expression, if you have a small number of cells and full transcript data, or you have a very large computational cluster, we recommend SCDE. If you have batch effects, you can use some of the fancier methods. If you have nice balanced batches, EdgeR and MAST work very well on those. If you have super large data sets and don't have a large computational cluster to deal with it, the Kruskal-Wallis test and Wilcox test are fast and work reasonably well.
What's what do you mean by super large for SCD, and what's the time frame we're looking at?
For SCDE, more than 1,000 cells is hard.
Hard on one computer or on?
On multiple computers. So what did I run? I ran 800 cells on 10 cores, and it took 6 to 8 hours. Yeah, so yeah, SCD is slow, but it's good. So if you can, use it. If you can't, other options are available. Yeah, overnight. Yeah, over a weekend. And then we have advanced exercises, or you can work on your own data sets, or come ask us any other questions. And yes, before you leave, make sure you fill out the course survey, which should be on all of your desktops. You can. I don't see any real benefit to doing so. It's
Experimental.
It's more it's sort of better if you want to investigate how well Spikeins work.
Yeah.
But if you're interested in your biological situation, it's not necessarily.
Just one more additional note. So the advanced exercise, you have a description what to do here. So we use the website, which is called Conquer, and there you can find a collection of single cell data sets which were processed in the same way. So there are only 18 of them, unfortunately, but they were all aligned and mapped in the same way. And the scatter objects were created, so you can download the scatter object from this website and start working on it using the course materials if you want to. Or if you have your own data set with you, you can ask questions and we'll help you. So here is a code that you can use for the Conquer website. Also, we have a page which is called Resources, and here we just put some links to some useful resources. And I think one of the main links is this awesome single cell collection. So this is a GitHub repository where you can find a list of all available tools for single cell RNA-seq data. And there are quite a lot of them. And it's updated by different people. So this is probably the most extensive resource on single cell tools. Yeah, so also we have our own website where we keep our data sets that we use in our group. So there is a link here at the bottom of this page as well. So here you can find data sets. They are split by the organism and the biological study. So you can look at, for example, mouse brain. And here you have a data set, link to the paper, some supporting data, and description of the data set, the scripts that were used to generate a scatter object, then the scatter object itself, and a report based on the scatter object, which is automatically generated for each data set in the same way. So this is some QC plots and PCA plot using the scatter. So the ones that you've seen yesterday and today. No, these are, so basically these are publicly available data sets. So it's not our own data sets. If you look at the scripts, so for each data set, you can figure out how we download the data. So this is probably not a good example. So for example, this one, this is also not a good example. So the problem is we try to download data sets from GeoArchive. For example, here, right? You download the data set, but then not all of them contain the cell labels. For example, in the paper, they report some cell labels, but they're not present in the GeoArchive. So we ask authors to provide the cell labels. They provide it, so we store them on our own server. And basically, but you can still find the scripts. And then there is an R script, how you create a scatter object. So you can actually go and look what we've done. But these scatter objects are just raw objects. We haven't done any quality control.